Welcome. Welcome to our Ask an Expert, Harmful Algal Blooms Along the California Coast, a science briefing for the California Legislature that is also open for a broader public audience. This is brought to you by the California Ocean Science Trust, or OST for short. My name is Heidi Waite, and I am a science officer at OST, and I'll be your MC for the day. Um, for those of us who For those of you who don't know us, OST is dedicated to accelerating progress towards a healthy, equitable, and a productive ocean future for California. We were created by state legislation to formally strengthen the bridge between this cutting-edge research and sound ocean management. We support and bring world-class science and innovation together with state and federal policymakers to foster ambitious action on critical ocean issues. As a nonpartisan organization, OST serves a unique role in supporting the California legislature and state agencies in the stewardship of our ocean resources. This briefing is brought to you by OST's Legislative Science Services Program, which brings science advice to lawmakers. But today I'd like to start by thanking our sponsors for this briefing, Senator Tony Strickland and Assemblymember Diane Dixon. We're very grateful to have your support on this briefing and would welcome your remarks as we begin our discussions today. So I'll start first by introducing Senator Strickland, who represents the Senate District 36, which includes coastline from Seal Beach to San Clemente. Previously, Senator Strickland served in the Assembly, Senate, and as mayor and council member of Huntington Beach. Born in Fort Ord, he is a proud Whittier College alumni, where he earned a BA in political science and received the prestigious Nixon Scholar Award. This past May, Senator Strickland awarded the Pacific Marine Mammal Center in Laguna Beach as Nonprofit of the Year for California Senate District 36 to recognize the center's dedication to ocean stewardship and all the efforts to rescue and respond to sea life strandings like sea lions, dolphins, and whales. So I think Senator already has your camera on and unmuted and ready to go. I'd love for you to kick us off with some opening remarks.
Well, Heidi, thank you so much. I really appreciate everybody taking the time to be on this call, this Zoom. We're going to talk about an issue that's very important to me, and I'm sure everybody on this call. I'm proud to co-sponsor this legislative briefing because we're talking about harmful algae blooms across the California coast. You'll hear today from 5 subject matter experts who will provide insight on this critical issue that we're facing here in California. I want to share why this topic is very important to me. I represent all the coast of Orange County, from Seal Beach down to San Clemente. I'm a lifelong beachgoer, and even more importantly, I'm a big animal lover. I was Humane Society Legislator of the Year. I think preserving our ocean is vital. I want to thank Heidi for mentioning that I did recognize the Pacific Marine Mammal Center in Laguna Beach as my nonprofit of the year this past year. They play a crucial role in the day in and day out to share our deep love for marine mammals. There's many disturbing instances of marine mammals washing ashore by harmful algae blooms. It's important for us to understand the reasons behind this. And right now it's very alarming what's going on. And we need to know the who, what, and why. That's why we're on this call. Since February 2025, the harmful algae bloom has spread across Southern California. For the 4th consecutive year, a major bloom is in our region. We've had, in March of 2025, more than 120 marine mammals, including sea lions and dolphins, were stranded in Orange County. 700 sea lions and more than 200 dolphins have been involved in these cases. And a lot of them, and I won't mention the exact, because I'm not a scientist, I'm not going to mention the exact acid, but these mammals are testing positive for these poisonings. And I want to leave it to the experts to explain, but just know as a state senator, when I see just last month, there was 13 poisoned pelicans, and I mentioned dolphins, sea lions. I think we need to do whatever we can as policymakers here in California and hear from the experts and find out why it's happening and what, if anything, we can do to prevent this from the future. Because again, as an animal lover and as a beachgoer my whole entire life, I think this is a problem that we absolutely need to know about and see what we can do to solve that issue. And so I want to hand it back to Heidi. Thank you for all your leadership, Heidi, and everything that you do. And I want to thank the team for joining this conference call and moving forward on this critical issue facing my district and also all of Southern California.
Thank you, Senator. We appreciate you being here. We're also thrilled to welcome our other co-sponsor, legislative co-sponsor for today's briefing, California Assemblymember Diane Dixon, to give some opening remarks as well. I'll do a brief introduction here for you, Assemblymember.
Tony's going to look at my face and go, "What happened?" There was nothing I could do about it.
Yeah, we appreciate you
Are
being
you
here.
okay, Diane?
I'm okay. I just an update on my skin cancer, which is a message for everyone to take good care of their skin. And this I was telling Heidi earlier when I logged on. Today at 7:30 was the only time the doctor was available during this entire recess to do this surgery today. So I'm fine. It's all good. But I got 4 more events today that I will have to explain myself to. So anyway, all good.
Thank you. Amazing. I'll give a quick introduction and pass it off to you. So Assemblymember Dixon, you represent the California Assembly District 72, which also includes coastline from Seal Beach to Laguna Beach. As a former mayor and city councilwoman of Newport Beach, she is dedicated to safeguarding California's coastline, and her leadership on issues around ocean plastics and pollution also really showcase her commitment to the coastal environment and its resources. So with that, I'll pass it off to you to say a few words before we jump into discussions and hear from our experts today.
Well, good morning, everyone, and good morning, Senator. So I'm delighted to be a co-sponsor with the Senator in participating at the front end of your conference. I am thrilled and honored because I, too, care a great deal, obviously, as I represent not the entire coastline, only 37 miles. So those are important miles. And if we don't have a coast, if we don't have beaches, if we don't have quality marine life, then Seal Beach, Huntington Beach, Newport Beach, Laguna Beach will only be called Seal, Huntington, Newport, and Laguna. No beach. So we economically and a quality of life as for preserving and protecting our quality water systems and our ocean and our harbors and our marine life is absolutely job number 1 for me. So I'm delighted to speak with you this morning. So the cities that I represent, Seal Beach, Huntington Beach, Laguna Beach, and then I go inland to inland cities, Lake Forest, Laguna Hills, Aliso Viejo, and Laguna Woods. So thank you, Heidi, and the California Ocean Science Trust for having me as a co-sponsor. I have only recently become aware of the fine work that your organization does. It's an exceptional organization that effectively bridges the gap between scientific research and public policy, which gives me great confidence in my legislative duties. If you are involved in the policy formation phase, I will continue to look forward to your opinions and guidance and scientific thought processes, because that's obviously what I need to be effective in these issues. And educating communities in coastal and marine preservation is vital. And your algae, or as you know better than I've just learned this in such depth in the last several years as I've been serving in the legislature and as an elected official, council person, and mayor of Newport Beach, how important understanding the organisms, the marine life in our harbor and our oceans, and algae being the foundation of our ecosystems. As you well know, they produce the oxygen we breathe and provide food for marine animals. However, when algae grow excessively due to nutrient pollution from sources like fertilizers and sewage, they can form what are known as harmful algal blooms, if I'm pronouncing that correctly. And these blooms produce toxins that pose a significant threat to the environment, delicate ecosystems, and the health of both humans and animals. A couple projects that I've been involved in for many years that relate to trash in our water systems, coming into our harbor systems, and ecosystems that relate to ultimately if we don't prevent them, trash going into the Great Pacific garbage patch, which is alarming to everyone who knows about it. And my purpose in life is to let everybody know about it. So one project that I'm particularly proud of, when I was first elected to the Newport Beach City Council and chaired the Water Quality Committee, so I had a deep dive education into the water quality issues, I endorsed, sponsored, led the development and acquisition and construction and financing of the Upper Newport Bay Trash Interceptor. And I've been part of this project since its inception over 10 years ago. My colleagues and I on the Newport Beach City Council and I learned that hundreds of tons of litter were flowing into the Upper Newport Bay. So we wanted to find a solution to protect against trash ending up on our beaches and then ultimately finding its way to the Great Pacific Garbage Patch. I'm proud to say that in my first year as an assembly member in 2022, 2023, I was able to continue my work on this project and secured $1.5 million from the state of California, which was then needed to complete the construction of the interceptor. The interceptor, from start to finish, took 10 years going through various state agency processes, our city council, then the Orange County Water District, then the Coastal Commission, then about 5 more other agencies. So it took 10 years. And then by the time they got to 10 years, they needed $1.5 million. And I was delighted to help procure that money for district support. And on its first day, when it was installed just this last January, the trash interceptor already made a significant impact, collecting 4 dumpsters of trash and debris floating in the bay. And it is expected to continue to collect up to 80 to 90% of the floating debris before it pollutes our city's bay and harbors and beaches. And because I mentioned about how important tourism or the economic aspect of our beaches, which is driven by tourism, so it's very important that my district is conscious of these factors because we rely heavily on tourism and our marine life relies heavily on a healthy ocean. So those are very economic drivers, and the marine life is very important to what I do in Sacramento. These are taxpayer dollars at work dedicated to making all of our beach communities better. Now, I just want to quickly update you on another project that is ongoing right now. When I was about 2 years ago, when we had major storms all over California, especially in Southern California, and the flow of trash down the San Gabriel River, a river that borders Los Angeles County in Long Beach area, and my district in Orange County. And as it happens, all the trash comes down, tons of trash, that's not an exaggeration, and deposits itself on the beautiful beach of Seal Beach. And I was doing a trash cleanup one particular day when the rain stopped, and the local officials encouraged me to come and see the trash because it all has to be removed by humans picking up trash and voluntary support, which is wonderful with the coastal cleanup organizations, which is really valuable. So I said to the Seal Beach mayor at the time and public works staff, I said, you need to talk to Newport Beach. We're in the process of installing this trash interceptor. We need to put a trash interceptor in the San Gabriel River. So I started a working group. I involved the supervisor at the time was a Supervisor Doe, interestingly enough. He's had a lot of publicity recently, but before all that happened in his wayward ways, he did allocate $500,000 of Orange County funds to fund the concept creation of a study for what do we need to do in the San Gabriel River, because the flow is much different than in Newport. So we formed for the last 2 years the San Gabriel Working Group, and I encourage the senator to be part of this. His predecessor was involved in this, Janet Wynn, who's now supervisor. So she's been engaged on this, as is the Assembly Representative Josh Lowenthal, who represents Long Beach, and other elected officials. And just last week, I met with Supervisor Janice Hahn of the Los Angeles Board of Supervisors, and she's very excited about this project. She is familiar with the one other trash interceptor, which is located in Bologna Creek and Marina del Rey, and Janice Hahn, that's part of her district, or was before it was redistricted, and so she cares very much about this. So where we are right now, We have a consulting firm that's doing the conceptual design, and we're nearing the end of a 2-year study on that, and it will be presented. It's a very different concept. We have a donation from Oceans First. You may be familiar with them because they have provided the funds for doing a trash interceptor, but we need about 6 to 8 other storm drain capturing mechanisms that this is what Los Angeles County is going to fund. So it's a bipartisan, two-county project to put this trash interceptor in the San Gabriel River, and all the elected officials from all of our respective cities and the counties have been working, and our staffs have been working very tirelessly to put all this together. There are 13 cities that feed into the San Gabriel River, and all these cities are supportive of doing their part. So it's really, really, really a wonderful project. I encourage all you to keep track of as well and support in any way you can. I've also met with Secretary of Natural Resources, Mr. Crowfoot. He's very supportive as well. So I'm focused on, I see Heidi's here, I got to move on, but I just want you to know that I join with you in partnership to help keep our water systems healthy, and whatever I can do, and conferences like this are very helpful. So know that my staff, we are all in on water quality and trash. So whatever you can do and marine life. I should say, I too went and gave a certificate or resolution to the Pacific Marine Mammal Center, which is outstanding. I learned all about what they've done with the algal blooms and the rescue of all of these sea lions, and saw how they've been working. They're an amazing organization, so I too commend their good work. So thank you for having me. I look forward to hearing some of your presentations, and thank you for all you do to help keep us educated to do the right thing for our oceans and marine life. Thank you.
Amazing. Thank you. And we appreciate you being here today and for all your work in the ocean space and the plastics pollution space. We really appreciate all that work you're doing. And want to just say thank you to both of you for your sponsorship and your interest in this briefing. We really appreciate you taking the time to be here. I know that you all have busy schedules and important things to do, so if you need to, feel free to sign off whenever that may be. In the meantime, we'll ask cameras to be turned off so we can make space for the other speakers. Thank you again for those opening remarks.
Well, have a great conference, and keep me on your list. I'd like to know what you're doing. Thank you very much.
Absolutely. Appreciate it.
Thank you.
All right. Well, we now have an excellent speaker lineup for you today. The next 40 minutes will be a series of lightning talks from our 4 speakers, followed by a moderated discussion. They're going to give an overview, high-level science perspective on the impacts of harmful algal blooms, or HABs for short. You're going to hear that a lot. They're going to look at the impacts on the ocean, coastal processes, and health. They will discuss the value of rapid response research and long-term monitoring in understanding and predicting those blooms. We're planning for about 10-15 minutes for Q&A at the end, so we encourage you to ask your questions throughout the presentations via the Q&A function that's at the bottom of your screen. The webinar will close at 12:15 Pacific time, but will be recorded for distribution afterwards, so keep a lookout for that link following. Before we get into presentations, I'd like to briefly introduce each of the panelists for the day, and then I will pass it off to our first speaker. Our first speaker will be Dr. Alexis Pasulka, who is a biological oceanographer and associate professor at California Polytechnic State University at San Luis Obispo. Her research focuses on understanding how and why marine microorganisms vary over time and space. Her lab leads data collection at the Cal Poly Pier Shore Station for the California Harmful Algal Bloom Monitoring and Alert Program, also known as HAB-MAP. You might hear a lot about this today. It is a network of shore stations along the California coast that monitor for these HAB species and associated environmental conditions. Our second speaker today will be Dr. Holly Bowers, who's a research faculty at Moss Landing Marine Labs, where she studies and monitors HABs in the Monterey Bay area as part of that HAB-MAP network. Her research focuses on techniques like molecular methods and imaging to detect those HAB species and the trends that they're seeing. She is a past co-chair for the National HAB Committee and has served as a subject editor for the journal Harmful Algae. Our third speaker will be Dr. Raphe Kudela, who is a phytoplankton ecologist and professor at UC Santa Cruz. His research focuses on how factors like light, nutrients, and physiology control phytoplankton growth and distribution patterns in the marine environment. His lab is also part of the HAB Map network and is a member of the Global Ocean Observing System Bioeco panel. And then finally, but not least, our fourth expert and moderator of the Q&A panel that'll happen at the end is Dr. Clarissa Anderson, who is a biological oceanographer and whose research focuses on the prediction of harmful algal blooms and toxins in estuarian and coastal ecosystems. She's also interested in the fate and transport of those toxins to deeper waters and sediments. Clarissa is now at the Scripps Institute of Oceanography, serving as the executive director of the Southern California Coastal Ocean Observing System, or SCOOS, and the director of the NOAA Cooperative Institute for Marine, Earth, and Atmospheric Systems. She is a chair of several different national HAB committees and a member of the science advisory team for the California Ocean Protection Council. So with that, I want to pass it off to Dr. Alexis Pasulka. I invite you to turn on your camera. Great. And I'll hand it over to you to give us a brief background on harmful algal blooms.
Thank you for having me. Hey, hopefully my screen is sharing okay. Great, yes, so I'll get us started with a bit of an overview of harmful algal blooms in California. So phytoplankton, which are the organisms that form harmful algal blooms, or as HABs, as you've been introduced to, are microscopic plant-like organisms that depend on light and nutrients to grow. There are many different types of phytoplankton, but collectively they play a critical role in the ocean as the base of the food web. So they're consumed by these tiny animals called zooplankton, which in turn are consumed by higher trophic levels like invertebrates and fish, and eventually top predators like us and marine mammals. Phytoplankton have really high growth rates. So unlike terrestrial plants, which can take months or years to grow, phytoplankton populations can actually double in days. And when they grow in really high numbers, they form what we call blooms. And some of these blooms, despite the microscopic nature of the individual cells, can actually be visible from the sky, as shown here in the photo of a bloom off La Jolla shores. Now, from an oceanographic perspective, because of these fast growth rates, this means that phytoplankton populations track changes in the environment really quickly. So as temperature or salinity or light or nutrients change, so might the phytoplankton community, because, as I mentioned, there are many different types and they don't all have the exact same preferences. So we see variations in terms of their abundance, or how many there are, and their composition, or who is there. And this influences our coastal ecosystems and how they function because of their key role in the food web. Now, unfortunately, a subset of these bloom-forming phytoplankton have harmful impacts. This is what we call HABs. Now, the harmful impact of different groups of phytoplankton varies. So some produce toxins that directly harm humans and macrofauna like marine mammals, particularly as that toxin bioaccumulates up the food chain. But not all HABs produce toxins. Some can cause harm to marine life based on other things like their physical structures. So for example, the spines of some species can cause damage to fish gills. And some cause harm simply by growing in really high numbers. This can change food webs because there's a different prey source available. Sometimes the collective impact of all of the microbial respiration associated with blooms can lower oxygen levels in the water column. Sometimes it's the sheer numbers can actually reduce light, shade the water column, and reduce light to seafloor vegetation. And other species might produce mucus or slimes that have other negative consequences. So there are a variety of different ways that HABs cause harm. So the collective consequences of these blooms include public health risks, marine ecosystem impacts, and economic losses for the fisheries and tourism industries. Now, in California, two species cause particular concern because they are reoccurring bloom formers along our coast and because of the toxins they produce. These are Pseudonychia, which is the diatom on the left, and Alexandrium, which is the dinoflagellate on the right. Pseudonychia produces a toxin called domoic acid, which is a neurotoxin and causes amnesic shellfish poisoning. And Alexandrium produces a toxin called saxitoxin, which causes paralytic shellfish poisoning. So these are particularly concerning groups of phytoplankton, and you'll hear more in a bit about some of the recent events, particularly involving Pseudonychia along our coast. But there are a number of other groups of phytoplankton that cause harm, and we monitor here for in California because some of them produce other types of toxins, or others are problems because of these other harmful impacts I've described. And so an important part of understanding HABs and mitigating their impact is by detecting them. And so we do this through monitoring. And one of those monitoring programs that Heidi mentioned at the beginning is our Cal-HAB-MAP monitoring program. And many institutions collaborate to cover the coast of California. And you'll hear next about some of the monitoring efforts and the newer technologies we use to track HABs. And the products of these efforts include a data portal and a bulletin, which work to make the data publicly available and integrate findings from a variety of efforts. So that's kind of what HABs are and what the harmful impacts that they have. But the big question then is what influences the formation of harmful algal blooms, and why do some species bloom when they do? So phytoplankton, just like plants on land, grow better when they have their resources like light and nutrients. But in the ocean, nutrients in particular can be spatially and temporally limiting. And when that limitation gets released, things grow. And California is situated in an eastern boundary upwelling region. So seasonally, due to the winds and the direction of the California current, surface waters get pushed offshore, and deeper waters, which are cold and nutrient rich, move into the surface waters where we have sunlight and phytoplankton can have their resources. And we see this upwelling signal in ocean data. So here's an example from the Cal Poly Pier with temperature on the vertical axis and month on the horizontal axis. And we can actually see the upwelling signal in the cooler temperatures in the spring along our coast. And we can see the phytoplankton response to this upwelling as well. Again, this is data from the Cal Poly Pier with month still on the horizontal axis, but now phytoplankton concentration on the vertical axis. And we see some of the greatest concentrations of phytoplankton, which are these bars, and in particular diatoms, which prefer these cold, nutrient-rich conditions during upwelling. So when these phytoplankton have sunlight and nutrients, they can grow. And this isn't a local phenomenon, but happens seasonally along the entire west coast of the US. So this is showing latitude now along the vertical axis and month on the horizontal axis with color as chlorophyll, which we use as a proxy for phytoplankton biomass. And those warmer colors mean higher phytoplankton biomass. And you can see a general trend of higher chlorophyll during that spring season when we have upwelling. But you might also notice that the timing and duration and magnitude of chlorophyll varies with different latitudes. And so the pattern is not the same everywhere. And so this is an important factor because there are many scales of variability that we need to consider that influence these resources that phytoplankton need to grow. So the California coast sits within the Pacific Ocean and is interconnected with other ocean regions. So we need to just consider how these larger basin scale cycles like El Nino or extreme events like heat wave alters ocean conditions. And along the coast of California, we see spatial variability in where phytoplankton grow and bloom. So this is now a chlorophyll map from satellites where those warmer colors still mean higher chlorophyll concentrations. And on this regional scale, in addition to sort of seasonal coastal upwelling, which I just described, there's other kilometer scale oceanographic features that form. So for example, eddies, which are essentially create swirls of ocean water, can form along our coast, and this then changes nutrient delivery to the surface. And then if we were to zoom in to 3 examples of HAB monitoring sites spanning California, from Northern California at the Trinidad Pier, or Central California, the Cal Poly Pier, or Southern California down at the Scripps Pier, these sites don't just have variations in their oceanography due to where they're situated in California, but there's other local scale processes. So they might be adjacent to terrestrial areas with different population densities and land use. They might experience different amounts of rain or runoff and river input into the coastal system. And of course, on all of our minds recently are wildfires, which could have a local scale impact. So all of these scales of variability influence light and nutrients of temperature, which in turn impact which phytoplankton bloom and when. And so with the ultimate goal of predicting and mitigating the impact of HABs, what we try to do is combine our knowledge of the ecology and physiology of different groups of phytoplankton with these scales of oceanographic variability while monitoring for HABs. Now, of course, that's their ideal, but there are limitations. There's gaps in our understanding of different phytoplankton. It's hard to measure all conditions in all places across all relevant timescales, and we simply can't measure everything everywhere. But you'll hear today about some of the incredible monitoring technologies we have, how characterization of events and tracking trends over time has given us insight into HAB dynamics, and the real power of integration of monitoring efforts to really understand these ecosystem level impacts. So I'll turn it over now to Dr. Holly Bowers, who will talk more about our monitoring efforts across the state.
Great. And Holly, I'll get your slides up here in a second.
Great, thank you. Okay, great introduction. Thank you so much, Allie. You really set the stage for the rest of these slides. So as mentioned, I'm from Moss Landing Marine Laboratories, and I'm going to talk to you some about the monitoring strategies that we have along the coast. Next slide. So you've heard about CalHab map already a couple of times, and here's a simplistic map here showing the stations along the coast. And the idea is that we go out once a week at these stations and we collect various samples for analysis. So we get a snapshot of what's going on along the coast during that week, and we can see
Oh no, I think we might have lost her.
If you like, I can take over.
Maybe that's best for now. And if she gets back on, she can take over again. Thank you, Raphe.
Yeah, absolutely. She might be coming back.
about that. I'm tuning in from so we go out and we take measurements, and all of the stations, type of microscopy counts for species. Have some stations. We take water quality parameters like temperature, effect we get that. We also take nutrient samples, toxin analyses, shellfish, and do some imaging. And that is all of the stations have the capabilities, and so sometimes that gets offset by expertise and what's available. Next slide.
Sure. Holly, maybe we could try with your camera off, just 'cause it's a little choppy in audio. We'll try that.
Is that better?
Oh. I don't think I can hear you either.
Can you hear me?
I can't hear you very well. It's very choppy. Potentially we can have.
Yeah, I'm happy to jump in.
Thank you.
And so Holly was just talking about the CalHab map stations. And one of the really critical things is that we don't just collect the data, but we make it available. And so all of the data is being hosted on a website called AirDAP, which is basically just a big database. And you can go in there and you can look at the variables yourself. We use them to run models. We're using them to make sure everybody is reporting things consistently. And so that's a big part of why we're so successful in California. Clarissa and Allie are part of SCOOS, the Southern California Coastal Ocean Observing System. The other one is CENCOOS, the Central and Northern California Ocean Observing System. And these two observing groups work together to monitor many, many things, including things like plastics in the ocean and nutrients, but also harmful algal blooms. And so all of that data is going into these big databases, and it's publicly available to anybody that would like to access it. Next slide, please. And so what we use this for is we're trying to keep an eye on what's going on in the coastal ocean and alert the decision makers, the public, the scientists about what's happening. And so a big part of that is tracking both human and animal health events that are associated with these harmful algal blooms. And so all of these groups work really closely with state agencies. And so that includes OEHA, the Office of Environmental Health and Hazard Assessment, State Water Resources Control Board, California Department of Public Health, and the California Department of Fish and Wildlife. And that's really critical because as scientists, we can provide the data, we can make the measurements, but we don't make policy decisions and we don't make decisions on whether, say, the fisheries are opened or closed. And so we provide that information to these agencies that have the authority to make those decisions. They take all of that information, they evaluate the situation, and then that's where they come out and you might see posting on a beach saying avoid contact, or there might be a shellfish closure, or as has happened several times now, the Dungeness crab fishery is either closed or delayed because of the danger of harmful algal blooms. Next slide, please. And so one of the things that Alexis touched on is that all of California is not the same. And so one of the things that we have to deal with is that the environment is complex, that there's various things happening all the time, depending on the weather, the oceanographic conditions, the specific area that you're in. And so this very nice image from a paper by John Ryan is showing Monterey Bay and illustrating some of the things that can be going on. And so the colors, the reds and the greens, are two different types of phytoplankton. And so in Monterey Bay, we often sort of have a retention area, sort of a quiescent zone on one side of the bay where you see all that red. And we tend to get a lot of dinoflagellate blooms there, true red tides, discoloration in the water. And part of that is because those dinoflagellates are strong swimmers. And so they form layers, making it difficult to identify them during some times of the day. And then they swim up to the surface to get sunlight and then swim back down at night to get nutrients. And so they can become really, really concentrated because they're actually aggregating, they're swimming together. In the southern part of that bay, where it's more green, that's an area that's strongly influenced by upwelling, that big #1 arrow. And there, that would be more like Pseudonychia and domoic acid, where it's really being driven by upwelling conditions, by river runoff, and they can change their buoyancy a little bit, but they're not swimming up and down like the red tide dinoflagellates do. And so even within Monterey Bay, we can have completely different conditions on one side of the bay versus the other side of the bay because of this combination of biology and the combination of the ecosystem responses. Next slide, please. And so this becomes really important when we talk about monitoring. And so you saw we have about a dozen sites along the California coast where we're monitoring pretty regularly, typically once a week. But this is from one of Heidi Bauer's papers from 2018, where we had a big Pseudonychia event producing lots and lots of domoic acid in both fall 2013 and then spring 2015. And what you're seeing here, the size of those blue circles tells you how much domoic acid was in the water. And the two big yellow X's are showing you where we normally monitor. And so one is at the Santa Cruz Wharf in the northern part of the bay, and the other one's at the Monterey Wharf in the southern part of the bay. And so if we only had data from those two X's, you can see that in 2013, we would have completely missed this very toxic event that was occurring offshore. As opposed to that in 2015, which was the warm blob, the marine heat wave, we had domoic acid throughout the bay. But if you look really carefully, you'll notice that yellow X up in Santa Cruz, there's really no bullseye there. It's a little bit offset from that. And so if the only thing we were monitoring was Santa Cruz Wharf, we would have said, oh, there's nothing much going on. There's not much of a domoic acid event. When in reality, it was some of the highest domoic acid we've ever seen in California. And so it really drives home the point that a couple of shore station monitoring programs are good. They provide long-term continuity, but we really want to get that spatial representation of what's going on, particularly offshore where the animals are foraging and we generally don't have very many observations. Next slide, please. There's also issues going on at the level of biology again, at the level of species. And so within a given species, say Pseudonychia, some of them are producing domoic acid, some of them are not producing domoic acid. Sometimes they produce a lot of domoic acid, sometimes not so much. We have geographical differences, and so they're not identical, even though they're the same species. And so an organism identified in Southern California may not behave the same as one in Northern California because it has slightly different genetics. We have what we call strain differences. So again, even though they're the same species, we would call it Pseudonychia australis, for example. One of them might be a super toxin producer, another one not. One might like warm water, another one likes a little bit cooler water. And so that biology just complicates the picture for us. And one of the sort of exciting things as a scientist, but also of concern, is that we're always finding new species, new types of toxins that are out there. And if we don't know they're there, then we don't need to look for them. And so a big part of it is just keeping up with what's actually happening in the water so that we can be as protective as possible. Next slide, please. And so some of the things that we use are pretty simple, pretty straightforward. And so Alexis showed you some nice satellite images of chlorophyll. And again, on the right, the reds are high chlorophyll, high biomass, and the blues are low chlorophyll. And so one of the things that we use that's super easy to measure is we can go out and just measure chlorophyll, which is the total amount of phytoplankton in the water. And so we can use that using instruments such as on the left, where it's being lowered into the water, or we can take water samples and bring them back to the lab and measure them in bulk. And so that allows us to make some measurements really rapidly, or even see them on a daily basis using satellites. And it gives us an idea of where we should be concerned. Next slide, please. If we want to get from chlorophyll, which is just sort of all algae, down to specific organisms, that's where we have to put a little more effort into it. And so classically, we stick it under the microscope and somebody identifies what organisms are there. More and more, we're using genetics. And so we can sequence the organisms and find out exactly who's there. And there's some really amazing new technology that's been around for maybe 10 years or so, where we can use these automated microscopes essentially. And so the one in the center is a little small box, and it's connected to a flowing water system, and it takes pictures of the phytoplankton as they go by. The one on the right that looks like sort of a can with a complicated thing next to it is something called an imaging flow cytobot. And it literally is at some of these stations. It collects a sample about every 20 or 25 minutes. It takes a picture of all the phytoplankton, and then we use machine learning and AI to identify them. And so we don't necessarily even need to have somebody looking at all the images. It can alert us using that machine learning that there's a bloom of something occurring that we should be paying attention to. Next slide, please. The next question, of course, is it actually producing toxin? And so having the cells there does not necessarily mean that it's toxic. And so that's the next step that we usually follow on with. And so again, we can use molecular methods, such as in the bottom center, where we can use things like antibody assays that tell us whether the toxin is present. We have some fairly new methods that can measure toxin over prolonged periods of time. And so that thing that looks like an embroidery hoop in the middle really is an embroidery hoop. And it has some resin inside it. That resin absorbs the toxins. And so we can get an integrated view of different toxins that are in the water. And on the right is one of the fairly sophisticated instruments that we use to say with absolute confidence that this is the toxin that's there and that's the concentration that's there. And that information is really critical because that's, again, what the state agencies are using to say, do we need to close a fishery or do we not need to close a fishery? Next slide, please. And so none of this is free. Some of it is fairly inexpensive. A lot of it is not inexpensive. And so what we need to do is we need to be continuing investing at the local level, the state level, and the federal level, including new technologies and methods and expansion of monitoring. And I think that one more slide, perhaps. I think that was it. All right, great. And so
That's it.
don't mind, I'm just going to go semi-seamlessly into my presentation so we don't get too far behind.
Thanks for stepping in there for Holly.
Absolutely. And so you get to hear me some more. Now I'm going to kind of tie some of these things together and talk about what the trends are, why all of California is not identical. And so talking about the patterns that we see. And so I want to start sort of at the really high level. And a question that we as scientists often get is, are there more harmful algal blooms? And it was a really nice study with a global data set that looked at multiple years of data. And they published this paper a few years ago and said essentially that once you take into account how much effort we're putting into monitor HABs, at a global level, we can't actually say that harmful algal blooms are increasing. However, and this is key, at a regional level, such as California, which is that WCA box that I outlined in red, and it's the chart on the right that's outlined in red, we actually do see an increasing number of harmful algal bloom events in California and Alaska over the last 25 years or so. It's kind of borderline whether that's statistically significant, but yes, in California, we actually are seeing more events happening over the last 25 years or so. We zoom into Santa Cruz Wharf, I'm using some of those methods we just talked about. Here we're looking at about 20 years of data from the Santa Cruz Wharf, which is part of that HABmap site. And you can see we look at a bunch of different phytoplankton. And what you're looking at, the reds and the yellows are, red is a lot of that species and yellow is not so much of that species. And then white is we didn't detect it at all in our weekly sampling. And I circled two of them that you've heard about, Alexandrium, that causes paralytic shellfish poisoning, and Pseudonychia, which of course is producing domoic acid. And what I want to point out is if you just look at this for a little while, you see that it's not always yellow, it's not always red, it's not always white. Instead, there's sort of an ebb and flow. There's a pattern to these blooms coming and going. One of those patterns that we can identify using Alexandrium, which we're not talking so much about today, but we can look at that and we can say, well, what causes the blooms and the toxicity? And what we see is that it's positively responding to temperature. And that temperature is positively responding to something called the North Pacific gyre oscillation, which is essentially the strength of upwelling, which influences things like nutrients. And so looking backward, we can say, oh, there's actually a connection in Monterey Bay. When the North Pacific gyre oscillation is in a particular mode and it changes about every 10 years or so, we would expect to see more of these blooms. A really nice analysis did the same thing for domoic acid, taking about 25 years of data. And Marco Sandoval Belmar published this about 2 years ago. And he looked at all of California, but in particular, he looked at 3 areas where we have a lot of data. And so these little pie charts that you see, the upper left is Monterey Bay, the upper right is Santa Barbara Channel, and then the bottom third of the pie is the San Pedro area. And what he found is across all of them, some things are consistent. And so domoic acid and Pseudonychia responds to upwelling regardless of where you are in California. And that comes back to what Alexis was telling you. Generally, it responds to silicate limitation and to nitrate and phosphate limitation. But if we zoom in a little bit and look at some of these climate scale things, we see that as an example in Monterey Bay, the North Pacific gyre oscillation suppresses blooms, while in Southern California, it enhances blooms. Similarly, if we looked at El Nino, which many people are familiar with, it enhances blooms in Monterey Bay, but it suppresses blooms in Southern California. And so that's getting into some of this complexity that not everything is going to be the same across all of California. And that's why we need observations and modeling that are specific to the region. We can take some of that and project forward in time. And so this is using the global climate change models and basically looking at where would we expect Pseudonychia and domoic acid to be favored. And so red means we wouldn't expect it, yellow means it's sort of borderline, and green means really good conditions for domoic acid events. And what we would expect if things keep going the way they're going is we're going to gradually shift Pseudonychia and domoic acid up into the Gulf of Alaska, and is probably going to become a little bit less prevalent in California simply because the conditions are no longer right for it. And so in other words, we're going to see poleward intensification of some of these events. And we actually see that happening. And so this is the Humboldt area. If you look at panel C, that's the Columbia River up in Oregon. And after the marine heat wave in 2014, we saw a big shift where before 2014, there was not a lot of domoic acid off Humboldt. After 2014 through today, it's become a new hotspot. It has conditions that are favorable for domoic acid production, which is obviously of concern given that it's a big fishery area. And so what I just want to leave with is two thoughts. One is that we're pretty good at looking backward and telling you generally what conditions were favorable for these blooms. We're not so good at telling you next month, is there going to be a bloom in a particular area? And something that Holly touched on that we're a little bit concerned about is we're talking a lot about Pseudonychia, but there's other organisms out there that we're concerned about as well. And with changes in climate, changes in the way humans are using the ocean, we don't necessarily know who's going to win and who's going to lose in the future. And so just to wrap it up, the reason it's so complicated is there's a whole bunch of things going on at the same time. And so we've got climate change, we have nutrients, we have wastewater discharge, we have biology, and all of that is coming together to give us the HAB trends and the responses that we're seeing. And so with that, I'm going to turn it over to Dr. Clarissa Anderson, who's going to talk a little bit about some of those responses related to human health and wildlife health.
All right, share my screen. Hopefully this is going to work, and you're going to have to put your seatbelts on because I'm going to have to talk fast. We got to get back up to speed. We have a lot of Q&A, beautiful questions coming in. I'm trying to answer them as they come in. And I will do my best to cover a lot of information, and I'm sure much of this is stuff you're dying to know about the animals and those impacts. There was some great overview already, so a lot of detail I won't have to go on. This will be the crash course and everything food webs from this point forward. And I think I don't need to cover the physiological responses. Ali did a great job telling you what's going on with these animals and why these neurotoxins are so bad for them. What I want to point out is, when we're thinking about animal responses in the shellfish, all the way up to top predators, there is a lot of periodicity. There has been a lot of interannual variability. We've heard this in all the previous talks. But we do know that going back into the 60s, we've had these events. There was a beautiful retrospective analysis done by a group from Scripps and UC Santa Cruz, led by folks like Mary Silver, who did some of the earlier work on domoic acid in the Monterey Bay. And that really showed that in the 60s, this big event that's completely, forever canonized in the birds was indeed a domoic acid event that was impacting these birds. So at least going back to the 60s, we know that Pseudonychia was out there. It was producing domoic acid. We have sediment cores going back 1,000 years that also point this out, that it was there. We have sediment cores throughout the 90s showing domoic acid and Pseudonychia. It's not a new phenomenon, but there may be some changes that we need to talk about. We know in the 90s that we had a big bird event, and there was a La Nina of 98 that followed the major El Nino of 97, was kind of that first time we talked about California sea lions, California sea lion mortality. And from that point forward, there were some really big events. And Raphe touched on the 2015 blob event. This was the largest recorded food web impact that we knew about at the time, may still be, considering the geographic extent with Santa Barbara to Alaska. And we had thousands of animals that perished across the food web. But then we have something more unusual happening now, which is that from the last 4 years, 22 to 25, we have seen this kind of consistent large marine mortality event in response to domoic acid. And I would also say that after about 2000, we started to see these things becoming annual. The blooms were annual, even if not large animals weren't stranding annually. We're certainly seeing more shellfish poisoning from that point forward, and or at least toxins in the shellfish, not necessarily epidemiological records saying that people were getting sick, but at least that capacity for bioaccumulation in the food web. So I just want to point out, Ali said this already, but we learned a long time ago in those earlier studies that anchovy are a really good vector for getting domoic acid through the food web from the lower trophic levels to the higher trophic levels. And Since then, we've done more work and we've used other tools like stable isotopes to say, what if you're looking at a weird year like the warm blob, where we had this absolutely epic historic event, versus a year that's going to be non-anomalous for domoic acid, but also a little bit warm. What do the food webs look like in those different years? And could that explain the variability in what we see in terms of the transfer of domoic acid? So in a year where there is some in the water column, but it doesn't seem to be causing the impacts that we might see. And one thing we're seeing is that there are differences in structure, in lengths of the food chain that can explain some of these differences and how effective a vector, something like forage fish like anchovy is. So there are years where the food web structure is primed to lead to this large mortality in the upper trophic levels or in these top predators like California sea lions. So in that 2015 or 2014 to 2016 warm anomaly into an El Nino situation, we know that there were large impacts across the food web. But it was kind of the first time we really thought about Dungeness crab from California to the Pacific Northwest. Dungeness crab closure, huge losses. We've seen this a lot every year since then. And I think this was an interesting time where this kind of came into focus for us. And it also came into focus that there are these geographic differences in terms of impacts. Whereas in Northern California, we're thinking a lot more about Dungeness crab, even rock crab, and bivalves. When we get into Central and Southern California, we're thinking a little bit more about California sea lions because that's where those dominant rookeries are and where their haulouts are. It doesn't mean the bivalve problem goes away. We don't have Dungeness crab down here, but we still have to monitor bivalves for toxins. And the California Department of Public Health, as mentioned, does a great job at that. Their mandate, though, is sort of focused around those bivalves and now crabs, rather than forage fish, other fin fish, not really regulated or monitored for algal toxins. And there is no legislative mandate to do that. So I just want to say that, these Dungeness crab impacts have continued year after year, and they have their own story in relation to things like the humpback whale entanglement. We're not going to talk about that here. And then California sea lions are really a strong sentinel for these domoic acid events. And I didn't mention this earlier, but this is really to say that a sentinel just means that if we don't catch it in our nearshore monitoring, as Raphe pointed out in some of Holly's slides, we often see these animals come in sick before we know that a bloom is happening or we detect it in our monitoring. And this is a pretty critical thing. But one of the things we do have is a synoptic way to look at these blooms using satellite data and using model data. It's been combined into a product called the California Harmful Algae Risk Mapping System, CHARM. You can see the sort of synoptic picture here. It's been pretty good at picking up marine mammal strandings, getting that spatial representation. And because we've had this last 4 years of blooms, 22, 23, 24, 25, what you're seeing here is an average layer at the surface of what that CHARM model looked like. It's not a perfect match to how many animals ultimately stranded or died in those events, but it is gives us a good sense of sort of the where the bolus and most of the activity was happening spatially with respect to where these animal strandings were happening. And I just, can point out that if you look at 2025, that was a really strong Southern California event with a lot of the activity focused around Los Angeles and Orange County and San Diego counties. And you can see that reflected here with the warmer colors really standing out in Southern California. So it is kind of the best synoptic view that we have. And as satellite technology evolves, I'm sure we will have better tools of doing this. But sort of going back to that 2022 event, which started the last spate of 4 years of mortality events, that was an interesting one. And it really covers the point for Southern California in the way that Raphe did in Holly's slides for Monterey Bay, in that we really never saw the largest size class of Pseudonychia or domoic acid at our peer sites. But we had a lot of animals coming in. It was well over 400 or 500 in the end. Because we had NOAA event response funds, we were able to get offshore and go to places like Santa Barbara Channel with the NASA-funded Plumes and Blooms cruises and find out that the highest domoic acid was offshore and subsurface where these animals were foraging. And you can see that here, how much Pseudonychia was offshore as well. And another thing we found out in 2022 with the help of those funds, with the help of Raphe Kudela's lab, as well as Kathy Lafay's lab at the Northwest Fisheries Science Center, is that we're now breaking records of domoic acid in these marine mammal tissues. We saw really high domoic acid in 2015, 2016 in the water, but not as high in the tissue as we're seeing now. And we broke these records with sea lions in 22, and then with long-beaked common dolphins in 23. We may have broken more records in recent years, but I think we've had a really hard time getting a lot of these tissues processed in order to find that out, and that's a funding constraint and some other more recent political constraints. But we've had more cetaceans stranding recently, so that's more dolphins, more whales, particularly in this last year. And even more attacks on humans, which is a problem that we really need to look at, which probably has to do with chronic overexcitation of the neurons for these animals that are getting hammered year after year. And these rescue organizations are truly overrun and understaffed, as we all know. So looking at 2025, this is actually a banner year for detecting things at the shore stations. It was on Christmas Eve that we saw a big spike in domoic acid at the Scripps Pier. And this was actually preceded by animals stranding and domoic acid detected in Baja. So as we were moving to La Nina conditions in the oceanographic environment in the California current, this was turning on and kind of moving with that La Nina wave north, and we were starting to see domoic acid spike at all these stations. It's a pretty dramatic set of spikes if you look at that time series on the upper left, and you're looking at 2025 all the way on the right. And then we started to see the animals coming in a little bit thereafter. So we did have a really good early warning, and we had an epic year for animal strandings. This has been alluded to throughout this whole webinar. A lot of cetaceans, and this is when SCU started to realize that we could come up with some new graphics for communicating a lot of these numbers in real time to all the different constituents that need to know about this. A lot of these animals perish on the beach. I just mentioned, attacks, they're on the rise, and a lot of these animals are eventually rehabilitated, but they restrand because they're so impacted by chronic domoic acid. So this is one of my last slides, just to say that we're really trying to get this communication built out. We're working with SCORP, we're working with Senkus, working with all the partners in California to share event response efforts, to get a whole synthesis of information out as quickly as possible, create the situational awareness that all of our partners need, especially because we have other toxins like saxitoxin, which is very dangerous and causes paralytic shellfish poisoning that is turning into a bigger problem. And this was the first year, as far as I know, where in California we were measuring domoic acid and saxitoxin in animal tissue. It doesn't mean that saxitoxin was the coup de grace. It was probably domoic acid in most of those cases, but it can't help. And it's something I think we need to keep a close look on. So this is my takeaway slide, a lot of words. And I think it's important to kind of review that we know a lot about adult female sea lions being a dominant sentinel for these events, but there's so much about food webs we do not understand. We do not understand entirely how things vary from bloom to bloom based on which forage species are there, the depth of those forage species. There are years where we only see seabirds stranding. Maybe those seabirds are more impacted that year because the anchovy or other forage fish are more at the surface. There have been years where we've only had shellfish impacts and no other animal impacts. And so these are the kinds of questions we need to get to. We need better tools for understanding that. We have satellite tools that have good fidelity with marine mammal strandings, but they don't give us an ideal forecast for all of the food web impacts. We do not monitor all of these forage species or top predators for these toxins. And we've now had a very unusual 4 years in a row with records being broken. Animals attacking people. We don't like to use the word attack, but I mean, in many cases, these bites we have no other language to describe them except really horrible encounters between wildlife and humans, multiple toxins threatening the animal health. And then we've got to think not only about mitigating these events, but how to communicate them and get people the information they need when they need it. And that's where I will end. Thank you so much. Thank you to California UST, and we will go to Q&A, and I will stop sharing my screen.
Yes, thank you, Clarissa. The other panelists, if you could also unmute and come on camera, we can move into our Q&A session for about 10 minutes or so, and we'll try to answer as many questions as we can. Just a reminder to submit those questions in the Q&A box, and we'll have until about 12:15, about 12:14, 13, and then we'll close up by 12:15. Amazing, thank you.
Okay, I am looking at our Q&A. Since I am the moderator here, I've got to get us on track to see what's been answered and what hasn't. I'm going to go a little bit here into the audience Q&A and pull a few things up. I know that I punted the offshore wind development question from Sherry Hafer. I don't know if you would like to get online. I don't think you can get on, but I will pose this question to our group. I think you can see it. I'm not 100% sure if you all can see it, but the question is really about any offshore wind development in Morro Bay and whether we have monitoring studies close to this area before, during, or after a project like this, which was in April 2024 to November 2024. So I will give that to Ali to answer.
So we don't have offshore phytoplankton data in that area. Certainly, there have been some studies looking at various different ecosystem impacts and trying to understand sort of the state of the ecosystem. We do have phytoplankton monitoring more near shore in Morro Bay, prior to and after this that would be directly related. But we will not be able, with the current data we have necessarily, to be able to detect the change in HABs events associated with offshore wind.
That's great. And thanks, Raphe, also for helping out with some of these other Q&A. I'm going to jump to Ellen Prager's question, which is, how do most people and decision makers get the information or access data, especially warnings and forecasts? A bit of that was touched on already. We do have websites. I don't think the CalOOS portal was mentioned yet, but it's a really great visual place. And we have the California HAB Bulletin, which I was remiss in spelling out, but it's a monthly bulletin to get a lot of this information to the public. Raphe, would you like to follow up on that?
Oh, no, I hit the button by accident. I'll just say a lot of it comes through the agencies. And so we provide information to the agencies, and then CDPH, for example, puts out the public notices. But Clarissa and Scoos do a great job of putting out approximately monthly summary of what's been going on. And that's on the Scoos website.
Thanks. And I didn't even see that you had done anything, Raphe. I'm not even looking at the Zoom. I'm in the questions. So yeah, the next one I want to point out, this is also from Ellen. Our funding impacts impacting our work, wet research, monitoring, or information transfer will be impacted. This is a doozy. We would need another half hour probably to get into this, but what I would just say is that so far we've been okay. A lot of this funding for monitoring does come through NOAA IUS, and we're seeming to get some of the research funding this year that we would have expected, but future years are in great, I would say, potential peril, considering what we're seeing with the FY26 president's budget. So that's where we're putting a lot of our effort, is trying to message and understand what could happen if these programs do go away in the next year or so. All right, so Danielle Bobrowski, I am curious to know if there's monitoring going on in Ventura Oxnard area. That is a great question. I don't know, Holly, are you able to answer questions right now? How's your sound? No, she's gone. Okay. So how about Ali?
Yes, absolutely. There is, I mean, again, what Raphe had mentioned is that we have point source locations along the coast at many of the piers in along Southern California and Northern California. So we don't necessarily have monitoring at every site along every pier in our area, but we do have monitoring near that area in Southern California.
Thank you. And I do want to read this comment from Lauren DeMaio. This is a nice comment. It's really more of a comment than a question. It's about the Dungeness crab shutdowns that can also impact whales. I did bring that up very briefly, but when we're looking at Dungeness crab season, it's also overlapping with a movement of humpback whales who've come in close to shore recently because of anchovy populations also being close to shore. And so she points out that with this entanglement issue, the work that's getting done would really benefit from more advanced warning, real-time monitoring, so that we can see the train coming before we have to, worry about whale mitigation. And I think that is a really great point. Marcus Emerson asks, or says, as a photographer who frequently gets in the water with sea lions, is there a resource for monitoring domoic acid outbreaks to hedge against getting attacked by infected sea lions? So, maybe some of the terminology here we could play with. Infected might not be the best way to think about it, but they are sort of intoxicated and impacted by a deadly neurotoxin that completely alters their behavior. And it doesn't necessarily make them more aggressive as animals, but they are suffering and they have an inability to control their physiology at times, which is what leads to a lot of this. And so the best resources we have are the ones discussed today in terms of the monitoring that we do, which is put out into the public domain, the monitor that the California Department of Public Health does. And then, of course, the kind of information that we're trying to get out there about marine mammals. And I did show the sea harm plots. Those are publicly available too, and they are a 3-day forecast for the potential for domoic acid poisoning along the coast. And it looks like a weather map and gives you that kind of spatial awareness and also temporal awareness. So if you have more questions on that, let us know. All right. I also wanted to bring up, in case I probably should go to Zoom and make sure no one's hands are up since I'm in the document, but I wanted to bring up a few things that have been in the registration questions, and I know that Ali touched on, and that is the impacts of wildfires. We don't want to ignore that at all, and we recognize that that is likely a very big component to what happened this year. I mentioned that this year's bloom in 2025 that went from about December of 2024 to May of 2025, so pretty historic in its length, was really focused in Southern California, but in its fullest extent, it did go from Baja all the way through Bodega Bay in terms of our detection of domoic acid and pseudonychia. But in LA, we do recognize that there was the potential for ash and other chemicals from runoff to impact these blooms. And so there are some studies going on. We don't have great understanding of that just from the monitoring alone. You would need to do some pretty in-depth experiments to see how those things can impact one another. But there are experiments happening in various labs, including, I think, in Raphe Kudela's lab and some labs at Scripps to better understand through experimental mechanisms how things like nitrate from ash or ammonium from ash or ammonium from fire retardants might enhance the ability of these blooms to grow. We don't see it as the causative factor, but we do see it as a possible enhancement. All right. So I do see Lorraine Sadler. I'm going to go to your question in the Q&A. It says, have read about the ability of Pseudonychia to bloom even if nutrient loads are low. So what in the diatom allows this to happen? Oh, Rafe's in the middle of answering that question, I see. Sorry, Rafe. I'm slow on the uptake. Do you want to answer it verbally?
Oh,
Uh-oh, I think
think
you're
you did,
muted
Ray.
still.
Thank you. Pseudomonas does really well at taking up all nutrients. And so upwelling usually brings up nitrate, but runoff and marine mammals produce ammonium and urea, and pseudonychia will happily use all of those. And so other organisms strongly prefer nitrate. And so when the nitrate goes away, they don't have much they can do, but pseudonychia can survive even when these low levels of nutrients are around.
Anyone else want to go with that? I know, Ali, you've done a lot of research on this too.
Yeah, I wanted to mention, I think it was answered a little bit earlier too, in terms of, again, I had mentioned sort of the seasonal upwelling, and that is often the time we see pseudonychia bloom. But one of the challenges is that there's a variety of other factors happening that may increase the toxicity expression, may change the duration and magnitude of some of these blooms. So we have to think about that in terms of, so we didn't mention sort of all of the other ecological interactions, competitive resource abilities, ability to outcompete for resources that really then influence whether or not pseudonychia blooms and then is toxic. And so, we sort of think of these seasonal cycles, but then imagine that a rain event amplifies nutrient loading simultaneously with seasonal upwelling in a region that could really impact the magnitude and duration of those blooms. And that takes time to unravel and understand. So as Rafe mentioned, it's often in hindsight, we look back and understand the magnitude of those blooms, and we're working at getting better at predicting and better understanding that.
Thanks, Ali. We had a question that I wanted to go back to from Brett Hartzman about whether it's possible to sort of mitigate these blooms with various technologies, whether that's vacuuming, skimming, any kind of different ways to increase or I would say accelerate the termination of a bloom. This is a huge topic in our field. HAB control is a big topic around the world. How to do it, how to do it with blooms that cover, thousands of kilometers and go very, very offshore and are also in the subsurface is a big problem for things like pseudonychia blooms in the California current. There are actually biological ways to do this with various organisms that might compete, but you can do a lot of this in a very closed system. It's very hard to do it in an open system like the Marine Coast. I think Rafe would be a great person to answer that further, so I'm going to push it to you.
Thank you. Yeah, it's as Clarissa said, it's difficult in an open system because things are always changing. And at some level, you have to be really careful about, large scale manipulations. And so where it has been effective in an open system at a small scale is in Florida. They did at one point test releasing viruses targeting the red tide specifically and showed that you could kill a red tide. There's been a lot of work globally and sort of in bayments, you can use clay flocculation, where basically it makes everything sink out to the bottom. But we have not had a lot of success at, a truly open system like California of stopping a bloom. If anything, some of the iron fertilization experiments suggest that we could probably start a bloom by accident. And so again, open systems are a completely different can of worms compared to controlling it in say a bay or in an estuary.
Yes, exactly. Thank you. Thank you for fleshing that out. And I think in the last minute that we have, I wanted to also just touch on seafood. There have been quite a few questions about seafood. We've talked about it here, and maybe just hit home the point that we do have a great network of agencies in California that are keeping us safe, keeping us healthy when it comes to shellfish consumption. But we do need to be wary, and we need to make sure the public understands risk when it comes to certain other fish that are not heavily monitored or regularly monitored. So we do have a lot of subsistence harvesters, people who love to fish off of piers, fish in the surf, things like surf perch. And these are animals where there isn't a lot of awareness of what's happening, and we're trying to get that out there. But when it comes to commercial seafood, when there is a large event like we had in 2025, you will see things like a sardine closure for the recreational and commercial harvest, which is what we had this year. But that is not very common, and I think it's good for us to keep this in mind, especially on the legislative side. So I will stop there, Heidi. I know we're at time.
Yes, thank you all for being panelists and dedicating your time and answering all these questions. I know we won't be able to get to every question today, but we did our best, and I think the panelists did a great job responding in real time in the chat as well. I just want to close out with a huge thank you both to our panelists for their time and expertise. Super interesting, very valuable discussion. I personally learned a lot. Just also offering our thanks to Senator Strickland and Assemblymember Dixon for their sponsorship and for joining us this morning. As mentioned in the chat as well, there will be a recording posted to our website. You can check the chat for that link, and I will follow up with an email as well through Zoom. Definitely get in touch if you have any questions or like any follow-up. Our contacts are on this slide here. And with that, I just want to say thank you for listening and take care. Thank you.